Hi, everyone. Thanks for coming. Thanks for sticking with us as we improvise a little bit. My name's Emmett McGregor. I am CEO of Sci-Fi Systems. We're an equipment provisioner for the processing space focused on hemp and large-scale cannabis operations. We do everything from extraction through end processing of ingredients, so isolate, distillate, et cetera. And I'm excited to talk about chain of custody as well as issues in sanitary and the definitions around what makes GMP equipment and GMP processing. I'm the odd one out in the group. I think everyone else is from the analytical side, so it'll be interesting to sort of compare and contrast ideas. Thank you.
Hi, everyone. My name is Annika Graham. I work at Untitled Labs as a chemist supervisor. We test marijuana throughout the state of Florida and hemp throughout the nation. I'm more interested in talking about standardization, full panel testing, traceability throughout the whole process, and basically any questions you may have, to be honest.
Hi, good morning, everyone. My name is Shari Campbell. I also work at Untitled Labs as a chemist. And in tandem with Annika, I also am interested in traceability, product efficacy, transfer of custodies, and safety.
Hi, I'm Antoinette Duncan. I am the quality director for CannaSafe. We're the largest cannabis testing laboratory in the state of California, and we're expanding. I'm directly responsible for our entire quality management system. I have about 13 years of experience in pharmaceuticals and medical devices, so FDA-regulated industries, and I'm bringing that experience in QA to the cannabis industry. So I'm obviously very interested in everything quality assurance and making sure that consumers are safe when they are consuming the products, but anything related to kind of application of, some FDA-regulated industry systems and processes to how we can apply it to cannabis, and also just in general analytical testing, standardization, I'm open to discussing.
Well, I have a question I was actually discussing with our moderator last night, and one that I believe he was really interested in getting answers from you all, and that is, how do you feel about protocol standardization versus proficiency standardization in the analytical space as it goes to federal standards across multiple regions and multiple industries? Are you comfortable with proficiency-based testing, and do you think that can be implemented effectively, or do you think that uniform protocols across multiple companies is going to be the future of analytical?
That's a good question. That is a good question. I think a good measure of both would be good. So proficiency testing to make sure that, because you can have protocols and they may not always, there's always a gray area where you can kind of finesse it to make it what you want it to be enforced to the past. But once you do proficiency testing from a third party, then there is no gray areas. Either you get the result that you're supposed to get at the target in the range that they allow, or you don't. So protocol will allow you to be standardized, allow you to follow a certain procedure so you don't stray from it. And I think proficiency speaks for itself. So people know that the results you're putting out is reliable.
And unbiased. It takes the bias out of it and the subjectivity. So when you're talking about testing, objectivity is the game, right? That's where you get trustworthy results and you get trust within your consumers and your manufacturers. And I think that's where this market, this CBD market should be heading towards.
I agree. I think that protocols takes away some of the creativity, I guess, away from the analytical laboratories. It kind of makes all of us the same, and that's not necessarily the case today. I think standardization is good to a degree, but you don't necessarily want copy and paste across the board. So I'm definitely a fan of proficiency testing. I think it's, like they said, unbiased, third-party evaluation of results, and it ensures that the labs that are participating in it are at least within the right ballpark, so that we're making sure that people aren't too far off, and it's just kind of an added measure of your processes compared to what should be out there, essentially.
Right. And to describe that in a little bit more detail for the audience, so the protocol standardization would mean everyone has to use the same process, essentially the same equipment to perform the tests along a prescribed line that would be regulated by the government. Proficiency testing is where as long as your testing methodology can test a standard, essentially randomly assigned sample to a certain degree of certainty, then whatever protocol you're using will be validated. So, and you'll be able to use that. So some examples of that is, HPLC and GCMS are commonly used in analytical labs today. There are some spectrometry and reference-based, light-based methods and protocols that are being used by some labs in the field, which are arguably much faster and inexpensive per test. So there's a big advantage for scaling up, but if it was protocol-based, probably you would have to choose one or the other, and that competition between technologies wouldn't be allowed to happen in the marketplace. So I think that's what we're talking about.
That's a good explanation.
Yeah.
So does anyone in the audience have any questions for us, specifically for your scope? Oh, sure.
Some of these? Sorry, can you speak to the availability of reference materials in cannabinoid testing?
Okay.
To what degree? So when you
Yeah.
say availability, do you mean the scarcity, or are you talking about the standardization of those reference standards? To what degree?
Yeah, no, sorry for the vague question. I'm talking about the breadth of standards that are available for both the cannabinoids themselves and terpenes and other things we're commonly asked to analyze.
So the availability of the standards. So right now, there's not that many companies making the reference standards that we need. And speaking on a Florida base, so we have Florida pre-drafted regulations out, and there aren't any standards made for residual solvents for that list. There's not one company that makes a standard that says, this is for Florida, you can use this to test it. So there isn't that much on that. Speaking on cannabinoids, there probably may be about a handful of companies that actually do make the standards. So the market is very scarce. And I think because we're now entering a time where we're actually, like, testing is being pushed. So I think the demand now for standards to be created is higher than it was before. So hopefully, as years go by and we continue to do more testing and people see the need for it, they'll make more standards. And even with the proficiency test, there are not that many companies that make proficiency tests for our market at all. So there is a need, and I think as it's growing, people start feeling that demand.
Yeah. And I think as more labs start to come into this market, the demand will start to increase. One of the issues that are out there right now is, so as Annika said, there's not too many reference standards or CRMs that are in the cannabinoid space or the cannabis space. And the other issue, and to compound that, is when you do proficiency testing, it's supposed to be something that is similar to the matrix. That's a whole nother ballgame. And it's something that we're struggling with constantly. So I think as supply and demand, as those things start to impart, you'll start to see more availability, and more availability specifically for the needs of this market in matrices, because as you know, matrices they vary. There's so
Hundreds
many.
of them.
There's hundreds.
Yes.
From toothpicks to
Candles
to
and-
Candles to
it's
candy,
almost
soda.
So it's
in everything at this
everything
point.
at this point. So being able to find CRMs that mimic or that resemble the matrix, meaning what it is, what it's been put in, is going to be an added problem that they have to work on for sure.
So to follow up on that, I think that it's really important when you're looking for an analytical testing laboratory for your products to make sure that they're validating in matrix. So there's a number of labs that will just validate to the standard, and it's really important to make sure that you're accounting for the variability that you're going to see when you actually test in matrix. And there, as we said, there's hundreds of different matrices. So sometimes you have to use a bracketing approach, like gummy bears are notoriously difficult to test, and there's all these other different matrices, so you kind of want to be able to pick some worst case options to kind of introduce as much variability as you can and make sure that you're still meeting your criteria for your validation. So that's something to look for in an analytical testing lab. The other thing I'll say is that if you're looking at these companies, some of these certified reference material companies, it's really great if we all reach out to them and let them know what our needs are so that they can start to put some of these things on their list. I know Cayman, for example, is a company who, they're really receptive to kind of hearing about what the demand is. And so if you're looking for a specific standard, reach out to them. I encourage you to reach out to them and make sure that your voice is heard so that they understand what the demand really is.
I think one of the things that some of the companies are moving towards are providing clean matrices. So they'll give you a clean matrix as in it doesn't have any spike of any of the analytes. And then they give you a spike that has the analytes, the reference analytes, and then you spike it yourself for homogeneity. Because that's another thing when you're testing for proficiency is homogeneity. That's incredibly important, whether it's you're testing a derivative anywhere to a brownie. You have to make sure that the sample that you're testing is a representation of the whole. To piggyback on what Antoinette said, reaching out to the companies that you're getting your PTs from or your CRMs from, speaking to them and telling them the challenges that you're facing, that might make change. Not might, because we in CACHA, it's something that we do actively. And we talk with our PT providers to see if they can help create solutions for the problems that we have, not just for us, but for everyone else in the industry that they provide PTs for.
Because whatever problems that we solve with them, that'll help every other lab. So the need for, as you said, proficiency test and a matrix is very important because that also shows your extraction, how well your extractions are doing. Buying those will then show them, hey, I might make some money off of making these proficiency tests. And more companies will start buying them when they put it out there to sell. And that's really big because you can understand, okay, I'm working with this lab and they do proficiency tests and they pass them. But if there aren't proficiency tests available, no one can buy it or show how well their testing really is.
And a follow-up question, you've been talking a lot about the diversity of different types of standards that you'd like to see in the market. For the main highest used standards, say standards for testing flour, for example, as the demand for hemp testing ramps up and the regulations are on batch size are changing, I know in Oregon we've seen a lot size of 30 pounds being brought into the hemp space for specific uses. There's ways to get around that and prove homogeneity across batches, but that's seen a huge spike. I mean, almost, I'd say maybe as much as 10x the testing year on year. Is there any risk that the standards will actually see a shortage, like not enough standards to serve all the analytical labs and the tests that are being done, or is it just more about the diversity of different standards that you'd like to see, that there's maybe not as much flexibility in the market yet?
That's a great question. As far as I know, the suppliers that we use, I haven't seen a shortage. We're always ordering constantly, maybe 10 packs, and we get them. I haven't seen it.
Yeah,
But-
we haven't really noticed a shortage either.
Yeah.
I think there's just, again, when you look at the number of labs that are in the cannabis space, there's really just not that many yet. And so I don't know that the demand is there, like we discussed before. Increasing that demand, increasing the number of labs that are out there will probably change that. There may be a shortage in the future, but so far, at least for the ones that we test for on a statewide basis, we haven't had any issues.
Is there a knowledge share for the labs themselves?
As in?
Do you guys collaborate with one another? Do you speak to labs, talk to other labs?
So we generally have proprietary information. So we create SOPs, and those SOPs are per our lab. They tell us our testing method, they tell us our extraction method. So as CACHA labs, we wouldn't go to CannaSafe and say, hey, CannaSafe, here are how we test things. But, and especially since we might be in different markets, but there is, if, for example, with the issue talking about proficiency tests, that definitely is a moment to collaborate and get the proficiency test providers on one scale, on what we need. But in regards to our methods, not so much. Although there are published methods out there that a lot of labs may use because they've been validated. All of your methods have to go through a validation process. They're in various places, so you can. I've seen them in PubMed. I've seen them in.
You type in Google, you probably see a good amount, but make sure your source is
sources,
good.
yeah.
So maybe
NIH.
NIH, PubChem, GC, LC, chromatogram, I believe. Just make sure your source is good, but they're out there.
Yeah.
So I'm a member of ASTM D37, which is the cannabis technical committee for ASTM, and there's a laboratory subcommittee as part of that committee, and we work together. A lot of different labs across the industry, we work together, and we're actually working to publish standard methods through that organization, so to encourage standardization across the industry. So, and it's not even just on the analytical side. There's also a processing and handling subcommittee. There's a quality management system subcommittee, which I'm on. And then, so there's actually 7 different subcommittees for different topics within the cannabis space. And so there's definitely opportunities. There's lots of organizations that are kind of, starting to pop up that are, encouraging different organizations within the industry to kind of get involved and collaborate. So I think that's, one opportunity, and that's a lot of times where we interact with other laboratories to kind of, work together and make sure that we're putting out something that we all agree with and we feel like is acceptable for the industry.
It's worth noting that a lot of the published methods are coming from the background of law enforcement. So, they're really used to characterize and identify sources of materials in the illicit market. So that has some advantages, but it also, in that there is published literature about it, but at the same time, their methods are perhaps constrained to what would be typical of a law enforcement analytical lab. So, yeah, there's definitely differences in the. I would say that most or maybe many labs evolve beyond those sort of standard methods very quickly.
Yeah, this is like a foundation. You start off when you go to school, you start off at kindergarten, and then you eventually get to 3rd grade reading, right? So you get your foundation, you learn your numbers, you learn your alphabet, and then you learn to form sentences and paragraphs and things of that nature. So you use it, as a foundation, and then you realize its constraints, and then you expand further. But that's a really good resource that you were just mentioning. That's a really great resource.
Yeah, it's a great organization. I mean, we are really collaborating. We're pumping out, standards left and right for the industry, and we're really hoping that it will be valuable, because I think we all agree that standardization is something that is lacking across the cannabis industry. And I know as far as analytical testing laboratories, I mean, we have clients, for example, who will jump ship and go to another lab just because they got a higher THC result, and it's like, natural variability for any method is, plus or minus 20%, sometimes. And so, a difference of, 2 ppm or, something like that for a specific analyte is really not significant. But, sometimes I think making sure that we have that standardization kind of decreases that, that likelihood where clients will kind of choose a laboratory based on the results, because we're all kind of like, at least for example, in the state of California, we have specific criteria that we have to meet, and all the analytical labs have to meet a specific criteria, as far as accuracy and reproducibility and those kind of things. So, so it's I think getting the word out about standardization and kind of how things work on the analytical side and the natural variability in the methodology is really important to discuss.
Absolutely.
So you brought up a big thing that I think a lot of people don't hear until later than maybe they should, which is the standard deviation of these tests. Could you maybe dig a little bit deeper into that, especially with regards to compliance testing where there's a maximum limit? Is the stated results in, say, a pesticide test or in the hemp industry in a THC test, is the stated result the midpoint of the range of deviation, or is it the maximum that it could be? How do you deal with those compliance limits? Could you elaborate a little bit?
So at Kaicha, if we get a failed test, so across all the tests, we do a mandatory retest. And the mandatory retest is to then confirm that failed test. So if you do get a failed test from us, it has been tested at minimum 2 times to reassure that that result is accurate. And we do a really good job with, if our clients ask for chromatograms, what was the prep, or anything, we share that information. So we try to be a glass door, glass window, so they can see, hey, these are chromatograms, this is the standard value, this is your value, this is how it matched up. If they want spectrums from the mass specs, we hand them spectrums to show that you may feel wary, but we'll provide you any information you need to be comfortable that this is ahead. And then we will speak to them and consult with them to figure out where this contamination came from and try to eliminate it with them. So, but there is variability in testing, but we do try to ensure that the number we do give out is accurate.
So I will add to that. I was reviewing the USDA hemp rules the other day, and I noticed that they actually provide a lot of specifics around measurement uncertainty and how you should interpret those results. And unfortunately, right now, there's not a lot of standardization when it comes to how to report your results and how to take into account the natural uncertainty built into the method. So it's really interesting how essentially what USDA has said is that as long as your result, the specification falls within your range of measurement uncertainty, then that's okay. So even if your result is, they're concerned about obviously THC percentage. So they're looking at, 0.3% THC. If you have a result that is, 0.35, you don't automatically fail if your measurement uncertainty is plus or minus 0.05, for example. And so it's really interesting to kind of see some level of standardization there that they've introduced, and they've really clearly laid it out in their hemp rules. And I think that would be really great to implement across the board, because right now, if you look at a lot of the testing lab certificates of analysis, most are not reporting measurement uncertainty with their results. So I think that that's something that we could improve on.
Definitely. And speaking of with hemp sterilization, I know the Hemp Council, what they do is if you are looking to make product and manufacture product, you can become a part of the Hemp Council, and they'll help you with all of that. They'll help you with making sure that you get through the right process, and they make you understand the variabilities of testing. And, because there's variabilities not just in your extraction method, there's variabilities within the instrument as well. And all those things are tested and verified. So if you're in the hemp industry, I know you talked about another one, but the Hemp Council is another one that can help, give answers to that as well.
I have a question. How you doing?
Hello.
Based on testing, I have a question. So let's say if we're doing white labeling or anything else, and they're doing the lab testing on their own and then shipping the bottles to us. And let's say on my own, I'm entering, I have retailers that I have my products on their shelves. Is there, depending on where they're storing it or how they're storing it, is there a level of variety of where the CBD content will diminish, where it will fall out of the of that area where it's like now on Instagram you've got a bunch of companies now that I go in, test it with third-party lab, and sometimes the lab test was from 6 months ago, and now like the CBD is dropped. It could not be from that. I just want to know like if it's something I need to worry about myself.
I could maybe speak to that a little bit. So shelf stability studies in CGMP environments are oftentimes done by the manufacturer themselves in other industries, especially in a CFR 111 nutritional supplement lab, and then potentially a third party could be engaged to do that. In the hemp industry right now, because GMP standards are, shall we say, spotty in adoption, there many products don't have shelf stability studies that have been done on them. And so it's going to completely depend product on product how long the CBD levels would stay stable, or any of the compound levels. And also, potentially more importantly, from a total supply chain standpoint, the actual shelf life from a consumer safety level, especially with regards to mold and other contamination that can build up over time for human consumable products. So that is an area of standardization that we hope to see a lot more adoption. And if you're in the business or getting into the business to start a company manufacturing products, or working with a company that's going to manufacture products on your behalf, a shelf life study, either in-house or preferably by a third party, should be a minimum requirement for your own products, but also for the vendors that you're relying on. So,
Shelf stability testing is critical, and it's just not done right now by many processors and manufacturers. I think that so I come from the pharmaceutical industry, and you mentioned dietary supplements and nutritional. It's absolutely required for 100% of your products in the pharmaceutical industry to have data behind your expiration date. Many of the products that we see don't have expiration dates, and if they do, they are completely arbitrary. There's no data really behind the setting of that expiration dates. Now, I'm not going to say everyone falls in that boat, but many of the larger players are starting to look into this because they understand that this is more than likely coming. I know the state of New York, for example, requires stability testing for their medical program. So there's certain states that are starting to kind of look into it and start to require that type of testing. CannaSafe, we actually just are starting our stability testing program. And clients are really interested in learning more about how does their product degrade over time, and what does my THC turn into over time, and what does my CBD turn into over time, and how long can I maintain my label claims? A lot of manufacturers and processors don't necessarily understand that about their products yet, and it's really critical because there is natural degradation. I mean, even from the way that you store not only the environment in which you store your product, but your packaging on your product has a huge impact on the stability over time. So there's a lot of clear bottles out there, with CBD and THC in it, and we know that cannabinoids are very susceptible to light. So I don't know if there's stability studies for all of those type of products that are out there. So I think it's really important for us to, as an industry, start to push forward in that and really have some data to support what's on the label.
Absolutely. I'll piggyback a little bit on that. In that, at Keitra, we're really big on making not just in efficacy, but in product safety. So with shelf life and stability, one of the bigger things is, do you have a microbial count that is starting to increase? And it's not necessary, and not all microbials, the presence of them are as important. Like, for example, aspergillus. Aspergillus itself might not be harmful. Some mycotoxins that they produce that are harmful. And so when you've put it in a clear bottle and you have it at not at ambient temperature or room temperature, are you contributing to the environment for sustainable microbial growth? So as a manufacturer, a white label, I would implore you to find a company that does shelf life and stability, one, for the efficacy of your product and for the safety of your product. Because at the end of the day, consumers are looking at you in terms of it has your company's name on it and not necessarily who you're white labeling from. And then in addition, when you get the product, you don't know what it was and what environment it was, how long it sat in that environment before it got shipped to you. So I would definitely find, are you in Florida? I would definitely find a lab that does that testing. At Keitra, we do that. And it's really, we pride on that. Anna and I personally sat for months making SOPs and testing it and making sure that the shelf life and stability is something that we can stand by, because we're big on safety and efficacy. And, the receptacle that you put your product in is, like Antoinette was saying, is extremely important.
Sherry said that your name is actually on the bottle. So when the consumers, maybe they get L from it, they're coming to you. They're not going to your white labeler. Right. So it's good to put, I don't want to say pressure, but to emphasize with your white labeler, we want stability testing, we want full panel testing, for your product, so you know that it's good.
And speaking about the manufacturers, this is actually a perfect time to talk a little bit about processing lines themselves. We're talking, we typically think about packaging as being, what faces the consumer, but there's another type of packaging as well, which is the wholesale ingredient packaging, and the storage conditions under which the ingredients are stored, as well as the testing that happens on ingredients before they go to the manufacturer to then be put into a finished good. Right now, we see very little standardization on the ingredient packaging side. We're seeing people use, 55-gallon buckets that are just purchased at wholesale from a general supplier that they may be intended for garden use or something like that. So really looking for packaging that, one, is guards against light, certainly, but two, is actually sanitary in terms of the surface condition of the packaging, because and this extends not only to the packaging, but also to all the surfaces that the product comes into contact to. So that's throughout the entire processing line. There's not yet a general standard across the industry for what sanitary means. It's different, for example, in the dairy industry versus the bakery goods industry, what sanitary means. So that's one of the big national level standards that has to be set in order for uniform GMP standards to be brought into the space. And our approach has been that we should meet the standards set essentially by nutritional supplements and pharmaceuticals as a starting point, so that we're reverse compatible with any future rules. And that means high polish, using only sanitary fittings, no threaded fittings for connecting piping that has product contact, and having the ability to do a complete clean of all equipment and clean in place procedures. So this contributes specifically to the shelf life and shelf stability of the end products, because it's what controls your initial microbial count and avoids having any contaminants in the product that will build up over time in the equipment being used to manufacture the goods. So you may see that, for example, your first batch of product is good and tests well with a vendor, but if they don't have good cleaning procedures and a sanitary facility, they may over time build up contaminants that you'll start to see in the product, and your shelf stability might go down. So going to a company that has GMP standards that are at this point compatible with either a specific type of food category that's closest to what they're manufacturing, or CFR 111 nutritional supplements, or most companies won't be buying from a 211 pharmaceutical facility at this point, because you can't really bring that easily back down into a consumer-facing product. So CFR 111 nutritional supplement standards, and looking for companies that not only know what that is, but have designed their facility around being compatible with those standards and implemented them, is very important. And you're talking about a very small portion of the total market at this point in the ingredient manufacturing side that have adopted that approach. Better adoption in the end consumer goods side, but also still not standard or uniform across the industry yet.
Yes.
How lengthy of a process would it be? And in ballpark terms, how expensive would it be to create for the first time a certified reference standard for a minor cannabinoid found in cannabis, so that its presence could be tested for and would show up in these certificates of analysis?
Can you elaborate when you mean a minor analyte? So generally CRMs or reference materials, they have an array of analytes that are generally found in that.
Yes,
Yeah,
and
okay.
just give you some context.
Okay.
I'm not in the industry and I'm not a scientist, so I'm coming from this from a very layperson's perspective. As a consumer, I'm checking out the different hemp tincture, hemp extract brands, and I see their marketing material and they talk about their extraction methods and how their extraction methods retain these naturally occurring substances, and then you hear that, oh, there's, upwards of between 80 and 120 cannabinoids and unique to cannabis that have been discovered thus far. But when I look at COAs, I see, there's like 5 or 6 listed there, and I'm like, where's that vast array that you're talking about? And then I've talked to some vendors about this, and they say, oh, it's prohibitively expensive to test for those minor cannabinoids. So that's kind of where I'm coming from in asking that question.
Okay, so I'll speak on 2 things. One, when you said that the COAs have a small amount of cannabinoids on there, I think that maybe that vendors should look into a lab that tests for a wider range. Because if they have like 6 or 5 on the listed, there's labs like Kaicha would test for 11. So we test for 11 cannabinoids. Also, cannabinoid standards are very expensive. And especially because there's not that great of a demand for them, they're going to be highly priced. So right now, to get a nice 11-part mix, you're almost looking at $1,000 for 1 milliliter. And that's that's really expensive. So yes, there are hundreds of cannabinoids out there, but I think it's going to take time for the market to grow, for there to be a demand for all those other cannabinoids, for them to make it to start testing for it. Because in order to sell the reference standard, at least for most companies, they'll go to an ISO 17034 or 43, and those accreditations are very hard to get. They're not very easy. And then to make sure that their standards are traceable, they're certified, it takes a lot. So right now, I believe the mixes have 11. I know there's a couple bit more, maybe a good 15 out there about. So I think it's really just for those companies to really feel the demand, to want to make more cannabinoids or put more cannabinoids in a mix.
The reality is that they probably haven't tested to meet that claim, because I don't know of a single laboratory that's testing for 80 cannabinoids.
Yeah.
And, honestly, even with ours, I think we test for, at least 11, maybe even 14. Sorry, I've only been at Canna for 2 months, so I'm still figuring it all out. But I think that, even some of our minor cannabinoids, if you look at our C of A's, most of the ones after THC, CBD, THCA, all of that, they're almost non-detected after that. So for you to go to even, the ones that are even more minor than some of the ones that we're already testing for, then more than likely, even if you were to test for them, they wouldn't actually be something that we would be able to detect in our methods.
They would be so small. Yeah, because, a lot of the testing labs, they test for what's the majority. And what a lot of research and support has been put into what the majority of those cannabinoids, how they affect our endocannabinoid system. So that's what they're testing. So those smaller ones, what Antoine is talking about, they're almost negligible compared to the CBDA count or the THC and THCA count. And our detection limits so when you are validating a method, you go through what's a method detection limit study to find out what's the lowest point that you can detect cannabinoids on, so that's the lowest point where you have certainty in a number. That takes time and a lot of money and a lot of reference materials that cost $1,000 a milliliter. So with all that, with combining all of that, it's almost
Prohibited.
Exactly, and it's almost it's nowhere cost effective, and testing for those minute cannabinoids without the compound amount of research that has been done on them, it almost doesn't seem, I don't want to use the word useless, but it almost doesn't seem worth the time and cost effective until there's a demand for it, or there's been an immense amount of research done by academia or independent laboratories to show how those minor cannabinoids, how they majorly affect our systems and the symptoms that we feel, and et cetera, et cetera.
And it's important to understand that when they say there's 80 or some would say 145 or 100 and X number of minor cannabinoids in the plant, the studies that have been used to characterize those cannabinoids are done by academic researchers in a context where they have access to analytical facilities that are not reference-based. They're using things like NMR, nuclear magnetic resonance, to characterize a new compound for the first time, but they're not doing the follow-on work to produce a standard for other labs to reproducibly test that in the field. So it's not practical for an analytical lab as a service to offer characterization of a lot of those minor cannabinoids. It would only be done on an in-house basis for a company looking to potentially manufacture an API, a pharmaceutical, for clinical trials, for example, to characterize some of those very minor cannabinoids, because of the issue that, in the field, 99 times out of 100, or probably more than that, 80% of those minor cannabinoids or greater would not be detected. There's variation in genetics and variation in the conditions the plants are cultivated under for those minor cannabinoids to be made, and in a general sense, there's almost always some of them in the plant, but which ones you would test for on a given plant, it's fairly chaotic in the market. There are, when you talk about cannabinoids and characterization of cannabinoids, it's important to note that isomers are included in those numbers. So when we say even THC, there's, I believe, 10 or more isomers of THC that are known. And when you're testing, typically you're testing for, acidic THC and delta-9 THC. But there are other compounds that have the same molecular weight or might test the same in a standard compliance test, that you would have to use very detailed methods of separation, of chiral separation, in order to be able to differentiate the different isomers. And again, in the broader industry, you only ever really do that in a pharmaceutical context where one isomer is known to have an activity that other isomers are not known to have. So I don't expect to see that in the market for general consumer products at any time, really.
Great question. Thank you. Thank you.
Thank you.
Thank you, guys.
Thanks,
Thanks,
everybody.
everyone.
Thanks.